All right, did anybody wonder why I put 2 lines here and 3 lines there? Really simple explanation, okay? Right here, when it comes to this nitrogenous base A and this nitrogenous base T, it only needs 2 hydrogen bonds to hold those 2 molecules together. Okay? So it requires 2 hydrogen bonds. But C and G, they're bigger molecules, and they require 3 hydrogen bonds to hold them together. So A and T only needs 2, and C and G needs 3. Okay? So here is DNA structure. It's made up of nucleotides. You have nitrogenous bases. A always needs to go with T, and C always needs to go with 3. Now remember that Tay-Sachs video. What happened here is instead of a G being here, maybe a A showed up. And that's a problem, right? That now this DNA is going to make a bad protein, and then that protein could not function properly in the body. Okay? So it's really important that when it is creating new DNA strands, that it always hooks up with the correct base pair. A with T, C with G. Okay, I want to scroll down just a little bit because those bases, you can tell which one they are based on the number of bonds. So if I have a molecule like this, and I have a mother molecule like this, and there are bonds connecting them. Okay, once again, the bonds in the middle, all right, these are your weak hydrogen bonds. All right, they can always be broken apart. So they're your weak hydrogen bonds. Now you tell me based off of the number of bonds here. So you have 1 bond, you have 2 bonds. What are the possibilities of nitrogenous bases? Okay, you have the choices A, T, C, or G. All right, so because there's 2 hydrogen bonds, we know that these molecules have to be A and T. Okay, and just for future reference, nothing you have to know right now, but if there's 2 rings of carbon, these are made up of carbon and nitrogen atoms, then that actually we know for sure this is a T, and this one, or sorry, is an A, and this one's a T. All right, but that's something you don't necessarily need to worry about. What I need you to know is if you see another molecule like this one, and for the sake of time, I'm just going to draw these solid lines. And there's 3 lines connecting it together. The fact that there's 3 lines representing 3 hydrogen bonds, then we know that we are looking at the molecules G and C. All right, with your partner, I just want you to quickly be thinking and talk about what is a nucleotide. Okay, this is what you should have drawn a picture of in your notes packet. Okay, what is a nucleotide? And what does it look like? All right, so we know that nucleotides make up DNA. And now we can pause the video and you can just review really quick what makes up a nucleotide. All right, so hopefully you discussed, we know it's a phosphate, it's a sugar, and what type of sugar? Well, it's a deoxyribose sugar, and it is a nitrogenous base. I'm going to go ahead and pick letter C. So if it is letter C, then what's going to hook up to letter C? Well, C and G always go together. How many bonds connect C and G together? 1, 2, 3 bonds. What type of bonds are they? Well, these are hydrogen bonds, right? They're really, really, really weak. Now, these bonds right here, okay, we're going to go ahead and highlight them because these bonds are strong bonds, okay? So those bonds are different. These bonds are the super strong ones, and they are covalent bonds. And unfortunately, these other bonds right here, all right, these bonds are the weak bonds. Okay? And those bonds are your hydrogen bonds. Really weak. All right, so let's build this complementary nucleotide. So there is guanine. It's always going to be hooked up to the sugar. And then that's hooked up to your phosphate. All right, do you need double helix? So A and T go together. C and G go together. It takes 2 hydrogen bonds to hook up A and T, 3 hydrogen bonds to hook up C and G. And between the phosphates and sugars, you'll always see strong covalent bonds. But between the bases, you will always see weak, weak, weak hydrogen bonds. All right, you guys should be good to go.